pharmaceutics, and then pharmaceutical chemistry, that is medicinal chemistry or synthetic chemistry, and then pharmacology. So in all these branches, how pharmaceutical analysis is playing its key role or functional role. The topic of mine is that. And then in pharmaceutical industry, what are the different branches where the pharmaceutical analytical techniques are used. As we have already gone to an industrial tour, we have seen AR&D department and quality control department. So I want to discuss what are the different R&D departments where analytical techniques are used, and what are different quality control labs or different departments in quality control department. And then what are the new analytical techniques that are used now in research and development. In various researchers are using this new analytical techniques, and how this pharmaceutical analysis is making research and development easy for the research scholars and scientists. So as we all know, pharmaceutical analysis is a branch of chemistry that involves in identification, separation, quantification, purification of substances. From a mixture of substance, it is used to know the determination of the structure or determination of the functional group. Mainly pharmaceutical analysis is playing a key role in quality and quantity of the products, and many research scholars, students, and then scientists in the industries are developing a method and validating the method, and they are using in quality control lab for routine quality analysis of the drugs. So what happens when quality control labs are using this method developed? They are increasing the efficiency, potency and quality of the drugs produced, thereby increasing the healthcare system globally. Once we are getting more potent, more quality and more efficient, efficacy drugs, then thereby increasing the healthcare system of the people. So we know the analytical techniques, they are broadly classified into two types: classical methods and instrumental methods. We know what is volumetric analytical methods. Those are complexometric titration method, redox titration methods, and diazotization titration method, non-aqueous titration method. These are all comes under volumetric titration methods. In gravimetric methods, we will analyze the quantitative analysis of the drugs or chemicals takes place in the gravimetric method of analysis. Instrumental methods, we know all the spectroscopic analytical methods like UV visible spectroscopy, infrared, NMR, then mass spectroscopy. Electrochemical methods, we know conductivimetry, amperometry, polarography. Chromatographic techniques, normally what we use is HPLC, GC. Then we have integrated techniques like LCMS, liquid chromatography with mass spectroscopy, and we have other techniques like X-ray diffraction methods in miscellaneous techniques. Hybrid techniques is nothing but integration of two or more analytical techniques is nothing but hybrid techniques. So in research development, how an analytical research development is taking place in industries or in the research level by the research scholars or the students or in the industry is mainly analytical method development and validation. Analytical method development and validation is nothing but developing a method and optimizing the method and validating it. Validation is nothing but keeping a documented evidence proving the developed method is precise, accurate, robust, it's rugged. We are giving an evidence that our developed method is all the things like precise, accurate and robust and everything. So we have to do a validation for every method that is developed. So this analytical method development and validation can be done by different instrumental analytical techniques like HPLC, LCMS, UV, and then GC for residual solvents. In that analytical method development and validation, we have stability indicating method. Stability indicating method is nothing but we will apply post degradation studies, like we will apply degradation like thermal degradation, alkali degradation, acidic degradation, and then temperature, pH. We will increase all these things and we will get the degraded products of our formulation or API. These degraded products can be analyzed by the method what we have developed. The method we have developed should be able to detect the degraded product by forced degradation study, that is stability indicating method development and validation. For one drug or one formulation we will use a single method development and validation. If we have drugs in combinations, for example paracetamol and diclofenac is in combination. In combination of 2 drugs or 3 drugs we will use simultaneous method development and validation. Simultaneous stability indicating method development and validation is nothing but we are giving the stability studies, that is forced degradation studies for simultaneous method development for 2 or more products. Then bioanalytical method development and validation, this type of bioanalytical method development and validation for single estimation and simultaneous estimation of drugs. In this we are taking plasma and serums of the animals or human serum or human plasma, and we are analyzing using different analytical techniques. Then drug impurity profiling, this is mainly industry oriented research where we will detect the impurities in the API or the formulation and we will try to reduce the impurities. I have already said I want to discuss the pharmaceutical analysis key role in different branches of pharmaceutical sciences. So we know in crude drug extraction, in a pharmacognosy we will extract the crude drug using Soxhlet, and then we will go for functional group determination, molecular weight determination, qualitative and quantitative analysis. All these are nothing but analytical techniques. Then in formulation preparation we will go for characterization of different formulations using analytical techniques. In pharmacological testing we will test the extracted drug on the animals, and then we will go for its optimization and concentration of drug by analytical techniques. And then in synthetic chemistry also we will synthesize the new drugs and we will go for the functional group determination, molecular weight determination, its quantitative determination by analytical techniques. And then in pharmaceutical industry, as I said, there are 2 main departments, quality control department and AR&D, that is analytical research R&D. We have 4 types of quality control departments, that is raw material quality control where the raw materials are analyzed like its physico-chemical properties, and then in process quality control where during the process, like for example we are taking tablets. Preparation of tablets by wet granulation involves different steps, weighing, sieving, then granules formation, granules drying, and then punching. So in between all the steps are evaluated. This is nothing but checked for its quality, that is in process quality control. Then packaging and labeling control, we have ampules, bottles, vials and rubber stoppers. All these are will undergo quality control tests. Then finished product quality control, where all the finished formulation will undergo quality control test. I said analytical R&D. Analytical R&D will develop a new method development and validation for existing drugs or for new molecules. Then this new method developed will be transferred to quality control department, and this transfer of new method developed is called as technology transfer. In AR&D department we will have 3 main departments, that is method development and validation department, stability department and impurity profiling department. Stability, what happens, they will store the drugs in 4 temperature zones with high humidity, high temperature, low humidity, low temperature, and then they will check the stability of the drug periodically, like for 1 month, 6 months, 12 months, 18 months, 24 months. Then formulation and synthetic, and these are the new analytical technologies in research and development. LCMS, that is liquid chromatography and mass spectroscopy, liquid chromatography and diode array detection, capillary electrophoresis and mass spectroscopy. Then GCMS, then supercritical fluid liquid chromatography, fluorescence, resonance, energy transfer, photon correlation spectroscopy, biomolecular fluorescence, complementation, transfer nuclear Overhauser effect, then yellow fluorescent protein-protein fragment, complementation, and surface plasma. These are the new technologies. Conclusion. So pharmaceutical analysis is playing major role. It is an interdisciplinary subject involving the other subjects, involving its roots in the subjects like physics, chemistry, then molecular biology and all. and then it is playing a key role in maintaining quality, efficacy, potency of the drugs produced in every preparation. Thus, analytical techniques is a subject of keen interest where the research development is done. Hence, this subject should be updated by the research scholars or scientists has on technology advances, so that the research scholars and scientists can produce more potent, more efficient and more safe drugs. Thereby the healthcare system increases, or we will have more expectancy of lifetime, and we will have more better healthcare system globally.
And all, good morning, madam. And now I am Shweta. I came from Gland Institute of Pharmaceutical Sciences. I am here to know to explain what are biosensors and what are the applications of biosensors, and what is the principle involved in the biosensors and working principle, how the biosensors work, on what principle they are working, and what biosensor consists of, what are the components present in the biosensor, and advantages, and what are the types of biosensors, and finally applications. The next, what is a biosensor? Before going to biosensor, what is a sensor? A sensor is a device which will sense a physical quantity present near to its device by an observer or by an instrument. Then what is a biosensor? Biosensor is the name itself tells bio plus a sensor. That means a biosensor is an analytical device which contains immobilized biological material and which is made up of a biological origin. So this is called as, so that's why it is called as a biosensor. That means the definition is an analytical device consisting of an immobilized biological material which can interact with an analyte and produce physical, chemical or biological signals which can be measured. Okay, and the next nowadays these biosensors are getting popular. Why? Because it's having several advantages. Before going to the advantages and how these biosensors are invented, and how, because these biosensors are invented by the hard work of the scientist called Professor Leland Clark, who invented the first sensing device, an instrument called Clark electrode, which used to sense the concentration of dissolved oxygen present in the patients, in the blood in the patients who are undergoing the surgery. First, he devised that Clark electrode. From later on, the research has gone, and based on his research, a modern, widely used glucose biosensor came into the picture. Then next, the basic principle involved in the biosensor is a biosensor consists of a immobilized biological material, a transducer, an amplifier, and then a detector. In this, immobilized biological material indicates that the biological material should be immobilized. The immobilization technique means we are making it a stability. We are making it a stable. Means we are undergoing some techniques like cross-linking, covalent bonding, entrapping the biological material to a support. Like this type of techniques are followed to make it as immobilization. Why we are going to immobilization means to enhance the stability, to enhance the reusability. So the type of immobilized materials like enzymes, living tissues, and whole cell microbials, antigen, antibodies are used as immobilized biological materials. And whenever analyte means a substance which is going to be determined, the concentration of the substance is going to be determined. That analyte, when come in contact with the immobilized biological material, the transducer, it will after biochemical reactions, the transducer will detect the product, and the transducer will convert the biological signals into electrochemical signals, and this electrochemical signal is amplified by the amplifier, and this signal is measured by our user-friendly devices like computers, displays, our graphs, like that. Then next, it is a basic principle what we discussed now, and what the biosensor consists of. Biosensor consists of two components, a physiological component and a biological component. In the physiological component, it shows transducer and the amplifier. In the biological component, it includes analyte and the bioreceptor. Analyte and the bioreceptor will, when a substance come in contact with the bioreceptor, it will check, it will detect the substance, and it also gives the concentration of the substance to the transducer. Before the transducer and the receptor, they undergo some changes, some production, it gives a product, it undergoes some physiological properties, changes in physiochemical properties of the product, and the transducer detects the products, and the transducer will changes the product, like the changes may be in the form of heat, light, mass change, absorption, light absorption, the changes may be. The transducer will convert the changes into the electrochemical signals that are measured by the detector. This is the biosensor. When analyte comes in contact with the biological material, it will bind to the biological material, and then the transducer will detect the material, and it will convert the electrical signals into the biochemical or chemical signals into the electrical signals, and finally the response will be observed. And this is the working of the biosensor. And what are the advantages? Why we are discussing this? Why these biosensors are getting more popular means the biosensor is small in size, so we can handle freely. So, and the next, and very small concentrations can be used, and the concentration of the substances are determined in complex mixture. In complex mixture, and these biosensors are used for a targeted specificity. That means antigen antibody have its specific antigen antibody binding. For that, we are going for this biosensors, and also no preliminary preparation is done for this, required for this biosensors, and they can measure the non-polar molecules that do not respond to the measurement of for other measurement devices. The next, what are the types of biosensors? Based on the transducers, based on the signaling devices, these biosensors are divided in several types, like calorimetric biosensors, optical biosensors, acoustic biosensors, and electrochemical biosensors. When next coming to the calorimetric biosensors are whenever the biochemical reaction occurs, they leads to a production of heat. It associated with the production of the heat, right? That heat it depends on the concentration of the substance which we are going to detect. So that, for example, estimation of the serum cholesterol. When cholesterol gets oxidized with the serum cholesterol oxidase, heat is generated, which is measured by this calorimetric biosensor, and when it is proportional to the concentration of the substance, right? Then next coming to the optical biosensors. Optical biosensors are those which will measure the light absorption, and also fluorescence or luminescence of the substances. And the best example for this is luciferase enzyme based optical biosensor. We all know that luciferase enzyme is responsible for the emitting light in the fireflies. Using this biosensor, luciferase enzyme based optical biosensor, we can detect the microorganisms, contamination of the product by the microbial attack. Whenever a product is having a microorganisms, it gets lysed. Okay, that lysed microorganisms can be detected by this type of optical calorimetric biosensors, luciferase enzyme based biosensors. The next coming to acoustic biosensors. Acoustic biosensors are those which having some crystals called as piezoelectrocrystals. These crystals change on vibrations. Whenever a substance come in contact or adsorbed on the surface of these crystals, it will detect, it will show some change in the frequency, means vibrations changes in the frequency, that will be determined by the use of these acoustic biosensors. Then next comes to electrochemical biosensors. Electrochemical biosensors, it also again classified into conductometric, amperometric, and potentiometric biosensors. Conductometric biosensors are used to know the changes in the conductance, electric conductivity and resistance of a given substance. Then next coming to amperometric biosensors. Amperometric biosensors are measured where the flow of electrons will be seen, means whenever there is a reaction which is having flow of electrons will be ions and electrons will be released. The flow of electrons lead to production of current. The production of current is proportional to the substrate concentration, and amperometric biosensor example is glucose biosensor, where where many people, it is a many widely used by the diabetic patients. And next, potentiometric biosensors. In the potentiometric biosensors, the potential difference is determined between the potential electrode, potentiometric electrode, and the reference electrode. And the example for this, ion selective field effect transistors, and which is used to monitor intramyocardial pH open during heart surgery. And the next applications, there are several applications in environmental, soil monitoring, food quality, where in environmental, like it will detect the harmful gases present in the air and the different types of harmful pollutants in the soil, like how the soil is degraded by the microorganisms. And then next, water quality management, what the toxins present in the defense, like in the military, all the soldiers are using these biosensors to detect the evolution of toxic gases. Any warfare activities are going on, they can be detected. And applications in different industry, food industry, for routine analysis, we can go for this biosensor also, because a small concentration is required for these biosensors. And then next, there are also nano biosensor which are used under nano bioscale, means at nano concentrations, or also we can estimate the concentrations. And these are the various applications of the nano biosensors. And current research is going on this.